So in this model, I'm going to derive the phenotypic covariance under the maternal GCTA model. Now, this is quite a scary looking slide with a complicated path model. So let's go through it slowly because it's actually a lot simpler than what you might first think. So on the left in blue, this is essentially one mother-child pair, pair I. And this blue model is exactly the same path model that we talked about in the previous set of slides. On the right in green is another mother-child pair. This is pair J. So again, it's exactly the same path model as in the previous slide. The only difference between what you're seeing now and previously is that we're explicitly including covariance paths between the two different mother-child pairs. So these can be paths that link the similarity between maternal genomes, and they're indicated by these path coefficients alpha. So you can see alpha between the maternal latent factors here and between here. So they're called alpha IJ because they link the similarity between the mum in pair I and the mum in pair J. And they're all called alpha because we're going to put them in a big matrix called A. But of course, there's also similarities between the children's genome as well. So these are indexed by these paths beta. So you've got a beta here, and you've got another one here as well. So we're going to be putting these beta terms in a big GRM called B as well. And then the last one is that we've got to consider the genetic similarity between mothers and children. So this could be either the similarity between mums and their kids in the same family. So previously we had set this path here to 1/2, but here set it to delta II. So that's actually the kind of empirical value on that genetic correlation. But we're also looking at the genetic similarity between mums and children from different families. So these are indexed by these delta paths here. So for example, this path here, delta IJ, is looking at the similarity between the mother in pair I and the child in pair J. Conversely, this path here, delta JI, is looking at the similarity between the mother in pair J and the child in pair I. So kind of the reverse of this one. And so these delta values are going to be put into a big matrix delta in the following slides. Okay, so let's have a go at deriving the expected covariance between the offspring phenotype in pair I and the offspring phenotype in pair J. So remember from your path tracing rules, what we want to do is we want to get from PI backwards along an arrow and then forwards to PJ. And we want to do that for all possible paths. So let's do that. So here's one that goes through the maternal genome. So this chain is M multiplied by alpha IJ multiplied by m. So we've got alpha ij m squared. That's our first path. But as well as the maternal genome, we've got a path going through the children's genomes. So this is C multiplied by beta ij multiplied by C. So we've got beta ij C squared for this path. But there's another one through the child's genome as well, this one here. So we've got H multiplied by beta ij multiplied by H. So beta ij H squared. So we can put those two, factor those two values. So let me just show you the previous slide there. So we're just taking beta ij as a common factor, putting it outside the brackets. We've got beta ij multiplied by C squared plus H squared in parentheses. Here's another path. This one goes through the mother's genome in pair I and through the child's genome in pair J. So we've got M delta ij multiplied by C. And here's another one, but this one goes through the child's genome in pair I and the mother's genome in pair J. So that's C multiplied by delta Ji, so the opposite, ij, multiplied by M. So again, we can do little factorization there where we take MC out of the parentheses. So you can see that. And this is the formula that we achieve. So here's the formula for the variance. And this is the formula for the covariance. And so you can see that they look very similar to one another. And in fact, we can write these in matrix form to get our M-GCTA model. So this is what it looks like. So it's basically very similar to the original GCTA model that we looked at, which only had two variance components. But now we've got quite a few different variance components. So this equation is saying that the expected phenotypic covariance matrix can be decomposed into a maternal bit, a bit due to the child's genome, a bit due to the covariance between the two, and a bit due to the residual environment. So A is a GRM containing all the pairwise genome-wide genetic correlations between the mums. B is a GRM containing pairwise genome-wide genetic correlations between the children. Delta is a little bit tricky. We'll go into that in the next slide in some more detail, but it's basically a GRM containing twice the genome-wide genetic correlation between mums and their children. And then I is the identity matrix. The thing I like about this parameterization of the model is that it contains what's called an implicit constraint. And that is basically that the covariance term can only be non-zero if both M and C are non-zero. And that kind of makes sense, right? Because if there's no maternal genetic effects, then you can't possibly have a genetic covariance between maternal and child effects. So, you know, if M is 0, then under this formulation, the covariance has got to be 0 as well. So that's a nice property of the model. So a lot of people, I guess, get a little bit confused about what exactly this covariance is. So it's just meaning that when you have genes in the mums that influence the offspring phenotype, when they get transmitted to their children, they also have some sort of effect in via the child's genome as well. So it could be an effect in the same direction, in which case it's a positive genetic covariance, or it could be an effect in the opposite direction, in which case it's a negative genetic covariance. So this slide just illustrates how we calculate the delta matrix. So we're basically just adding two n by n matrices together. And it's just literally adding a matrix and then the transpose of the same matrix. So if you imagine here, this matrix has the genetic similarity between mums and their kids, where mums are indexing the rows and the children are indexing the columns. And the second matrix is just the transpose of that. So now it's the children that are indexing the rows and the mothers are indexing the columns. And so when you add those two together, you basically get this delta symmetric sort of matrix. So there is another way to formulate this model, and this is the way that you could do it in the GCTA software package. So basically, rather than having that implicit constraint that we talked about in the previous slides, this is the formulation under the GCTA model. So you just have you know, your 3 or 4 GRMs here, one for your mums, one for your kids, the delta matrix and the identity matrix, and you just estimate 4 different variance components where you don't have that implicit constraint. So the nice thing about OpenMX is that you can fit that implicit constraint. GCTA software package, you can't do that. In simulated data, it doesn't seem to make a difference. I wouldn't go out on a limb and say that it doesn't matter in real data. So that's always something to be a little bit wary of. You don't want to sort of have models where you have a non-zero variance component for this covariance here, whilst either the maternal or the children's component is 0. That just kind of wouldn't make sense. So that's something to watch out for a little bit. And so this slide's just showing some empirical results from the original M-GCTA paper. So what we did is we fit the M-GCTA model to data from the ALSPAC cohort. So this is a big population-based study in the southwest of England, consisting of a couple of thousand mother-offspring pairs. And so what we did is we tested the model on 2 phenotypes. The first phenotype we looked at was kind of I guess a positive control. What we did as the offspring phenotype, we actually used maternal height as kind of a dummy offspring phenotype, in that if you think about it, mother's height, you know, is not going to be influenced by the offspring genome, but should be influenced by the maternal genome. And certainly when you fit the model, you get quite a strong variance component for the mothers, as you can kind of see in this table. So M is the maternal component, G is the child's component, Q is the covariance, and E is the residual component there. And so you can see that, the best fitting model under parsimony is one that just has a maternal component and a residual component in it. So that's kind of a nice positive control, I guess, for the M-GCTA model. We also looked at birth length. So this is obviously a perinatal trait. So you would expect contributions from both the mother's genome and the fetal genome, perhaps. Basically, we didn't really have power to resolve, definitively into these different variance components. But certainly, if you look at the full model, there seems to be some evidence there for a combination of maternal and fetal genetic effects on the phenotype. So I'm just going to finish off and give a shout out to some recent work from Espen Moen and colleagues at FHI in Oslo in Norway, where they've basically taken the M-GCTA model and they've extended it to look at paternal effects and assortative mating as well. And so they've called this method Trio GCTA. So if you're interested in having a read of that, that came out in Behavior Genetics in 2021.